How's it going, guys? My name is Tuli Mwarumba, and the topic of discussion for the long talk and the journals that I'll be discussing for this long talk will be about ocean acidification and its effects on marine life. Before we get started, I kind of wanted to explain the reason in which I chose ocean acidification and its effects on marine life. And one of the main reasons I did this was because in marine ecology, the class that I'm taking as well right now with Professor Johnston, we started discussing and looking into a lot of articles talking about acidification and its effects on the ocean. Excuse me. And before the class, I didn't really know much about ocean acidification except the fact that it was affecting coral reefs. And with my way of thinking, I somehow just believe that, coral reefs were not too important to the extremely biodiverse and extremely fragile ecosystem that is the deep sea, which is the ocean as a whole. And going through this class, we began, through these discussions and these articles, I realized how important coral reefs are. And so we learned a lot more about it. And so, what piqued my interest now was its effects on, marine life, its effects on not only, say, coral and, algae and things of that sort, but what does it do to, say, you know, predator prey, or what does it do to, trophic systems, or what does it do to this animal, and what can that cause? So that's what I'm going to be talking about. So let's get to it. A little bit of an outline. This is what I'm going to be talking about, and in this order. The first thing I'll be doing is talking about ocean acidification, what it is, kind of giving you guys a brief crash course into ocean acidification and its effects and what causes it. Then we'll talk about the effects of acidification on a sea snail that I'll discuss some more about later, and I'll give you some more details about it. And then the effects of acidification on bamboo sharks. So right there, I'll give you a little insider news. Like, this is kind of two organisms on different scales or opposite sides of the spectrum when you look at, predator prey, with bamboo sharks being considered a prey species in their system, and sea snails are known as, some are predatory. Are prey, excuse me. And then afterwards, I'll be discussing these and giving you some information about what this can cause or what might be happening due to these effects. All right, so let's get into it. So what is ocean acidification? So what is looked at the most when we look at the chemical components of the ocean is the pH. So as you can see on that bottom picture, the pH scale runs from 0 to 14, with 7 being neutral. And the higher it goes up the scale, the more basic the level of pH is, and the lower it goes from 7, the more acidic it is. And that, will go into turn with the ocean. So the ocean's pH is, on average, right now is at 8.1. The ideal pH level that we used to have, or the ocean used to have, was 8.2. And although this might not seem like a huge decline or a huge issue, it's a 30% increase. And with the ocean projected to become more and more acidic as the years go by, this is something that needs to be taken with serious, this should be taken seriously. So increased levels of CO2 causes the ocean's pH chemistry to become more acidified, or acidic, excuse me. So ocean acidification occurs when CO2 is absorbed into the water and at a high rate. It reacts with water molecules to form carbonic acid. And then this compound then breaks down into a hydrogen ion and bicarbonate. And this excess presence of all these hydrogen ions is what in turn causes the ocean to decrease in pH, thus making it more acidic. So why is this a big deal, you might ask? Shell dissolution for one. If you see in that picture, those are shells, and you can see that some of them look a little bit more sturdier. Some of them look like they're absolutely mangled and torn up, or extremely brittle. And this is caused by acidification, right? Because of this, because of less carbonate ion bonds and excess hydrogen, this results in fewer carbonate ions available for calcifying species to maintain their shells. So you see these shells, they're being destructed because there's not enough building material, let's say. And so as ocean acidification increases, the faster the shells dissolve. So the shells aren't as sturdy, thus making them more susceptible to break, erosion, and many other things. And that's a negative impact on the development of a marine organism. And then coral bleaching is one that I think a lot of people know, is one of the ones that is mostly in the mainstream, or most in the mainstream, should I say. And that's when coral are exposed to acidic oceans. They expel the algae that live within their tissues, which leave them bleach white. Although this may not kill the coral, it does have a drastic change, a drastic impact on the energy and the consumption of the coral. All right, so now we're going to be talking about the first journal that I looked at. And in this journal, it talks about, and it's looking at shell condition and survival of the Puget Sound pteropod. This journal was published on August 27th of 2014 to the PLOS One journal. I hope I'm saying that correctly, PLOS1 journal. And the authors of this, scientists Shailene Bush and Michael Mayer and Patricia Thibodeau and Paul McEllen, they look at Limacina helicina, which is a group commonly known as the small, it's a small sea butterfly. They're known as sea butterflies, which are small marine snails that have an aragonitic calcium carbonate shell and an important prey species in many marine food webs. So this is it right there. They're called sea butterflies, and you can see kind of this wing right here. I think you can see my browser. But yeah, and then this is the shell, right? So it looks really majestic. It's real translucent. It's really cool. And so you might be asking like, okay, what is aragonite, or what is this aragonitic calcium? So aragonite is one of the more soluble forms of calcium carbonate and is used widely by marine calcifiers, such as organisms with calcium carbonate structures, which is this snail, as you can see. So corals and other calcifiers are more likely to survive and reproduce when they have sturdier shells, and when this aragonitic calcium is in a good abundance, or the saturation state. As you can see down here, the aragonite saturation state is at a greater level, and we'll talk about aragonite saturation state in the next slide. But in this journal, they tested whether sea butterflies experience shell dissolution and altered mortality rates when exposed to high CO2 and a low aragonite saturation state. I cannot say that right now. I have no clue. Here it goes. So methods. So in this experiment, scientists collected adult marine snails that had an average shell diameter of about 1.94 millimeters from the Puget Sound, which is located in Seattle, Washington, right? And you can see this right here. Do you see how this straight, you can see this straight right here. And that is where water feeds into the Puget Sound. And what is a negative impact of this water surging from this current is that this California current, the water in this current has a low pH and it feeds into the Puget Sound. And so just take that into account, and it'll come back later. So in this experiment, like I said, they collected adult marine snails And they performed a 5-week-long experiment in which snails were collected and then put in 4 different controlled carbon chemistry conditions. And in this, they used a recirculating seawater system, and you can see there it's extremely complex in order to make sure that the environment resembled the pH and the aragonite saturation of these 4 controlled chemistry conditions. Okay, and so for the 4 conditions that they did the current conditions of during the summer surface, during the current conditions of the summer in the deep water. So the surface was 1, the surface water levels, the deep water, and then they did surface conditions during upwelling, and then also future deep water and surface conditions during upwelling. So the future one is what is expected through calculations and studies that have been done in other papers. And then after this, they measured the shell condition using a 5-shell characteristic. So in order to see where the dissolution or the erosion occurs, they tested it with 5 conditions, 5 characteristics to see if there was erosion of some sort. Opaqueness, the transparency of the shell, the brownness or scarred structures, and the corrosion and number of perforations that you see on the shell. So in short, if the shells were beat up pretty well, and if the shells looked as if they were brittle, this is how they tested it. So let's get into the results. I know it might look a little bit complex with all these dots, but hopefully I can make it not as scary looking after I explain it. So in this figure, it shows that the sea butterfly's survival as a function, the small colored circles is the fraction surviving within an individual condition. So with the experiments that are warmer colors, those were the shells that were that were picked up in May, in the May experiment, and then those of the cooler colors are the July experiment. The open diamonds is the fraction of sea snails that survived at the mean, and the small black circles with the vertical bars that you see here, the small black circles right here, that was the expected value, or so the expected value, the expected value of survival. And that was with the 95% confidence interval that was calculated from the best fit discrete mixed effects model. So from this data, you can see that the aragonite saturation state, it says aragonite saturation state on the x-axis. And so a little bit of background as well is that when the aragonite, it has been understood that when the aragonite saturation state is at a lower rate, there's less chance of survival. There's more stress on the shells, thus causing these shells to begin to erode. Once this aragonite state leaves 3 and goes lower, this is when the shells begin to erode and dissolute. So within this, you see that the aragonite saturation states are at 0.5, 1, and 1.5. So these are very low. These are very low states. And what this data suggests is that these shells are extremely sensitive to aragonite saturation state, especially below conditions of 1. And this shows it well because it shows that low aragonite saturation rates have really low fractions of survival, right? So this, and you can see this with the dots, right? So the fraction alive right here in this area with the low saturation rate of 0.5, you see less than 0.2, a fraction of them alive. And you can see this throughout the graph. And then also, if you see this picture to the right of this graph, you can see that the shells are extremely brittle, and this is within a 1-week process, right? So significant shell dissolution in the snails after 1 week of exposure kind of shows that low increase in acidification can lead to a decrease in aragonite saturation states. And what this does in turn is destroy these shells, which are very important to these snails. And the reason for this is that these shells are used to protect themselves. These shells are used to, mate and in order to continue the healthy upbringing and the healthy lives of these animals. Shells are extremely important, and not only shells that, as you can see in table C, like that's not helpful. We need to, for these animals to survive and to go through these adverse weather and through predator-prey relationships, in order to protect themselves, they need to have strong shells. And because of low aragonite saturation states, there's a lower fraction of survival rate of these animals because they cannot protect themselves as well as they could due to increase in acidification. So the second journal that I looked at were the effects, discusses the effects of ocean acidification on the white spotted bamboo shark. And you can see it right there. It's on the smaller side of sharks. And it's kind of, a very cool and interesting looking shark. And this journal was published by the Marine and Freshwater Behavior and Physiology Journal by the Taylor and Francis Group in 2018. Scientists Pegado, Santos, Cotoa, Pinota, Lopez, and Denise and Rosa were the authors of this and the scientists that took part of this experiment. And in this, the present study aimed to investigate the effects of ocean acidification on the growth, not only the growth, the swimming performance, and also the cholinergic system of juvenile white-spotted bamboo sharks. And so the only bullet point I got on for this slide is acetylcholinesterase, also known as, or you can, in short, ACHE. And what ACHE is the key enzyme in the nervous system of animals, which terminates neurotransmission at the cholinergic synapses, right? And that's one of the things that is being studied. So in short, I mean, after a neurotransmitter, to explain this a little bit more about what acetylcholinesterase does and what its job is, basically after a neurotransmitter molecule has been recognized by a synaptic receptor, it's released back into the synaptic cleft. And once this happens, it must quickly be removed or inhibited or inactivated in order for it, for prevention of constant stimulation and of the postsynaptic cell and excessive firing of action potentials. So basically, if this isn't inhibited, it continues. So to say this synaptic cleft, it must quickly be removed, because if this molecule that's been recognized by the synaptic receptor continues to be firing, it's basically doing a job that it's not necessary and it is not needed anymore. And so ACHE, if like the inhibition of ACHE can induce the loss of nerve conduction ability, causing hyperactivity, loss of coordination, convulsions, paralysis, and other kinds of behavioral changes. And I think that's one point that will go on to this next slide. So hold on to that one if you didn't get anything else. And so for the methods here, eggs were hand collected and transported, and once hatched, and once they were hatched, they were put in separate tanks randomly to a control or a simulated scenario of high CO2 for 45 days. So they were in these tanks for 45 days after they were hatched. And in this 45-day, their weight and measured body length were recorded immediately after they had been hatched, and then after the 45 days of exposure to their said conditions. After those 45 days, the sharks were transferred to a swimming tunnel with the same conditions they had just had in those 45 days in the tanks. In the swimming tunnel, swimming tests were had. In the swimming tests, you might ask, they wanted to observe the maximum velocity that they could reach and their burst potential and the amount, how often they burst. And in order to do this, water flow was increased at a rate of about 1 centimeter per minute until the shark could no longer swim and was considered exhausted. Whenever the shark stopped swimming, they would be encouraged to start, and in order to determine if it was just resting or if it was in fact exhausted and can no longer go. Chase protocol. So this was performed in order to determine ventilation and in distress. So what this did was when the animal was removed from the swimming tunnel and chased in a bucket by hand until it couldn't swim anymore. And then when it couldn't swim anymore and was considered exhausted, when it couldn't go anymore. After this, 5 days after the swimming tests were had, the sharks were humanely killed, and their brains were micro-dissected in 7 different areas. The areas are the telencephalon, which plays a role in most of our brain activity and is more analogous to the entire division of the nervous system. The cerebellum, which is responsible for muscular coordination. Optic lobes, that interprets retinal information. Olfactory bulbs, which receive neural input. The diencephalon, which is sensory and which sends sensory and motor signals to the cerebral cortex, and the brainstem, as well as the spinal cord. So what was found? In this test, it was found, as you can see in how you see it in figure 1, is that the effects of high CO2 on maximum reach velocity and percentage of time, that's what it shows. The effects of high CO2 on maximum reach velocity, the percentage of time swimming, and the number of bursts. On figure 2, it shows the impact of acetylcholinesterase activity on the 7 different micro areas of juvenile white-spotted bamboo sharks. So in this, both maximum velocity and number of bursts were not significantly different between treatments, but the percentage of time spent swimming was significantly lower in the animals exposed to high CO2 conditions. And that's the one with the asterisks right here. In comparison, you can see a large difference. And then for the figure below, it shows the ACHE activity decrease in 2 of the 7 brain macroareas. Analyze the telencephalon, which plays, like I said, which plays a role in most of our brain activity, and the optic lobes, which is part of the brain that interprets retinal information. So what this can propose is that ACHE activity, like in the previous slide, how I said it induces the loss of nerve conduction and ability, causing hyperactivity, loss of coordination, and all types of behavioral exchanges. It's argued that such findings may be associated or may be related to the decrease of acetylcholinesterase in this area of the brain, especially the telencephalon, which has been linked to motivation, right? So with these swimming times being drastically different and these ACHE activity decrease in the telencephalon and the optic lobes, it is hypothesized that there's a big correlation between those 3 things, due to the fact that the telencephalon just plays such a major part in the somatic system of animals, and especially these white-spotted sharks. So take-home messages. If you guys got anything out of this, I want you guys to know that ocean acidification affects all trophic levels, not only predators and not only prey, right? And not only does it affect these 2 things. I don't want it to be, for it to seem like it was only these 2 animals that it affects, but ocean acidification affects all trophic levels, and due to, say, sharks' inability to swim, that affects its ability to mate, its ability to lead, to protect itself from bigger prey species, and this can cause such a trophic cascade, and this can cause such an issue to other levels of this ocean biodiversity and this oceanic ecosystem. Another point is reduction in available carbonate in marine water adversely affects marine life with calcified shells. So acidification is destroying the shells of these marine animals. And what this in turn does is make it more susceptible to prey, more susceptible to just a lot of adverse effects that can come with it. And then the last thing that I want you guys to take home is that high CO2 also affects the growth and percentage of time that sharks spend swimming. And what this does is, like I just said, with the trophic level discussion, is that this can have a big issue on, or a big effect on the levels of marine life that are under these sharks, right? And also the sharks itself. With predator-prey relationships being changed by ocean acidification, we need to look more into what might be the cause of this and what can we do to stop it. So thank you.